Hello, and welcome to your Microbiology Bio 203 lecture on viruses, viroids, and prions. I am Mr. Kennedy, and I'll be guiding you through this material today. The first thing I'd like to do is introduce you to the general characteristics of a virus. Viruses are considered obligatory intracellular parasites, which, simply put, means that they carry on none of life's basic functions outside of a host cell. Viruses contain DNA or RNA, but not both. They contain a protein coat and occasionally a lipid envelope, no ribosomes, and no ATP generating mechanisms. If you look at this table, you can compare a virus to a bacterium and ascertain what one has and the other lacks. Hosts. Viruses can entertain a broad spectrum of hosts. There are all manner of cells, plant and animal, even bacteria that viruses can infect. Most viruses infect only specific types of cells in one host, not every cell. The type of cell that a virus infects is determined by specific host attachment sites and cellular factors. Bacteriophage are viruses that infect bacteria. Viruses range in size from 20 nanometers to 1,000 nanometers in length. Using this picture, you can get an idea of the scale of size of viruses compared to bacteria and even a human red blood cell. Next, we're going to consider viral structure. The viron. A viron is a complete, fully developed viral particle. It possesses a nucleic acid in the form of DNA or RNA. It can be single or double-stranded, linear, or circular. There is a capsid, which is the protein coat made up of capsomeres or subunits to envelop said nucleic acid. Occasionally, an envelope made out of lipids, proteins, and carbohydrates that coat certain viruses, and spikes or projections from the outer surface. This picture will give you an idea of some basic, generalized viral structure. General morphology. Viruses come in a wide variety of morphological shapes. There are helical viruses, which are hollow and possess a cylindrical capsid. Polyhedral viruses that have many-sided capsids. Enveloped viruses, whose capsid may be enveloped in lipid, protein, or carbohydrates. And complex viruses, which often have some combination of the previous three items. This would be an example of the morphology of a helical virus. This is an example of the morphology of a complex virus, commonly referred to as the T4 bacteriophage. Taxonomy of viruses. The genus name of a virus ends in the word virus. The family name ends in viridae. The order name ends in ales. Viral species are groups of viruses that share the same genetic information and ecological niche or host, but they aren't always exactly the same shape. Descriptive common names are used for species, and some species are designated by number. More often than not, viruses that share the same ecological niche will create the same symptoms and other types of outward expression in the individuals that they infect. Isolation, cultivation, and identification of viruses. Viruses cannot be cultured outside of other living cells. In order to culture viruses, they must first be introduced to living tissue in one way, shape, or form. Bacteriophage are grown in bacteria. Bacteriophage form plaques, which are clearings on a lawn of bacteria on the surface of agar. Each plaque corresponds to a single virus and can be expressed as plaque-forming units, or PFUs. This is a picture of those plaques. In living animals, viruses can be cultured. Viruses can be cultured in embryonid eggs. Viruses can be injected into the egg. Growth is signaled by changes or death of the embryo. This is an example of such an activity, where viruses can be injected into different layers or areas within an egg and cultured. In cell cultures, tissues can be treated with enzymes to separate cells. Virally infected cells are detected via their deterioration, known as cytopathic effects, or CPE. Continuous cell lines are always used when growing animal viruses in laboratories. This is an example of how that might be done. The tissue is treated with enzymes to separate the cells. The cells are suspended in a culture medium. The normal cells, or primary cells, grow in a monolayer across the glass or plastic container. Transformed cells, or continuous cell cultures, do not grow in a monolayer. Viral identification. Viruses do not leave us much to work with when it comes to classification. Therefore, we use their cytopathic effects, serological tests, Western blot, and their nucleic acids in order to classify them. Viral multiplication. Viruses multiply inside of host cells only. It must take over the host cell's metabolic machinery in order to replicate. There are two types of replication that viruses participate in. One is the lytic cycle. The other is the lysogenic cycle. The lytic cycle occurs when a phage causes the lysis and death of the host cell. The lysogenic cycle occurs when phage DNA is incorporated into the host DNA, but immediate death of the host is not always the case. There are steps to the lytic cycle and lysogenic cycle that you should be familiar with. In the lytic cycle, the first step is that the virus will attach to the host. Phage will attach by tail fibers to the host cell, and it can do so either externally or internally. Penetration is second. The phage lysozyme opens the cell wall, tail sheath contracts to force the tail core and DNA into the cell. Third is biosynthesis. Once the tail core and DNA have entered the cell, the host cell will then start to produce the phage DNA and proteins. Next is maturation. Maturation occurs as the phage particles are assembled into live, if you will, or active viruses. After that, the virus is released from the host cell. The host cell will simply rupture, spreading the virus particles throughout the environment or the body that possesses the host cell. This is an illustration of the lytic cycle from start to finish. The lysogenic cycle is a little different. In lysogeny, the phage remains latent. That means that the phage DNA will be incorporated into the host cell DNA, and it may stay there dormant for days, months, or even years. While dormant, it will be passed on to all future generations of that cell. At some fixed point in time, the phage will, for whatever reason, become active, and the lytic cycle will ensue. To get an idea of what this looks like, let's look at this diagram. In this case, you can see that a virus has attached to a host cell. It has also injected its genetic material into that host cell. Once that genetic material is in the host cell, rather than being expressed immediately, such that the lytic cycle would ensue, that piece of genetic material from the virus is incorporated into the host cell's DNA. At that point, we have what is considered a prophage. That prophage will be passed on to all future generations of that cell. This can continue for days, months, or even years. Then, at a fixed point in time, the prophage may excise from the bacterial chromosome and become active, initiating the lytic cycle. The steps of the lytic cycle that ensue are identical to the aforementioned process. Specialized transduction occurs when specific bacterial genes are transferred to another bacterium via a phage. Changes in genetic properties of the bacteria can result. In specialized transduction, we can actually use this idea of viruses inserting their DNA into a host cell to carry out the transformation of microorganisms to ultimately create organisms of our own choosing. This is just one way that we can use genetic engineering or biotechnology to solve the problems of society. Multiplication of animal viruses. When animal viruses reproduce, the virus can attach to the cell membrane. Entry by receptor-mediated endocytosis or fusion occurs next. The virus will uncoat. Biosynthesis follows. Maturation, where the nucleic acid and capsid proteins assemble, filling the host cell to the point where it may explode or have the virus released by budding. Biosynthesis of DNA viruses. DNA viruses replicate their DNA in the nucleus of the host using viral enzymes. They synthesize a capsid in the cytoplasm using the host cell enzymes. Unfortunately, for all cells infected with viruses, they lack the ability to detect foreign versus self-DNA, and any DNA present in the cell will be expressed. This is an example of the replication of DNA in an animal cell with an animal virus. Biosynthesis of DNA viruses with the Adenoviridae. Adenoviridae are the double-stranded DNA non-enveloped viruses. Adenoviridae tend to cause respiratory infections in humans and tumors in animals. Poxviridae are the double-stranded DNA enveloped viruses that cause skin lesions, vaccinia, and smallpox viruses. They are included in this group. Herpesviridae. Herpesviridae are double-stranded DNA viruses that are also enveloped, and they come in several varieties. Here you can see herpes simplex 1 through 8 and their related illnesses. Papaviridae is a double-stranded DNA non-enveloped virus. The example that you need to know is papillomavirus, which causes warts. Papillomavirus can transform cells and even cause cancer. Hepataviridae are double-stranded DNA enveloped viruses that cause hepatitis B. They use reverse transcriptase to make DNA from RNA. Viruses can multiply in host cells' cytoplasm using RNA-dependent RNA polymerase. RNA-dependent RNA polymerase comes in two forms: a sense strand, where the viral RNA serves as mRNA for protein synthesis, and an antisense strand, where the viral RNA is transcribed to a plus strand to serve as mRNA for protein synthesis. Occasionally, you will have double-stranded RNA. This is a picture illustrating both of those processes. Picornaviridae. These are the single-stranded RNA plus strand viruses that are non-enveloped. Enterovirus, rhinovirus, and hepatitis A fall into this group. Probably the one that you know best is rhinovirus, as it causes the common cold. Togaviridae. This is the single-stranded RNA plus enveloped virus. Alpha virus, which is transmitted by arthropods, and rubavirus, which causes rubella, belong to this group. Rhabdoviridae. are the single-stranded RNA minus viruses with one RNA strand. Lassa virus, which causes rabies, belongs to this group. Reoviridae are your double-stranded RNA non-enveloped viruses. Reovirus and rotavirus belong to this group. Single-stranded RNA viruses that produce DNA use reverse transcriptase to produce DNA from a viral genome. Viral DNA can integrate into the host chromosome as a provirus. Retroviridae, or lentivirus, like HIV and oncoviruses, do this. This is a picture illustrating the action of retroviridae. This table shows the families of viruses that affect humans. You have the characteristics, viral family, important genera, and clinical or special features listed column by column for your consideration. Finally, we'll consider viruses in cancer. Several types of cancer are caused by viruses. Many develop long after viral infection. Cancers caused by viruses are not contagious. Sarcoma is cancer of connective tissue. Adenocarcinomas are cancers of glandular epithelial tissue. The genes in question when speaking about cancer are our oncogenes. Oncogenes transform normal cells into cancerous cells. Oncogenic viruses can become integrated into a host cell's DNA and trigger the action of oncogenes inducing tumors. A transformed cell can harbor a tumor-specific transplant antigen, or TSTA, on the surface and a T antigen in the nucleus. Oncogenic viruses include adenoviridae, herpesviridae, poxviridae, papaviridae, and hepadnaviridae. RNA oncogenic viruses include retroviridae. Retroviridae is viral RNA that is transcribed into DNA using reverse transcriptase, which can integrate into host DNA. HTLV1 and HTLV2 cause adult T-cell leukemia and lymphoma. This concludes your lecture video on viruses, viroids, and prions.